So, delighted to present some of our work we're doing on gene-directed metabodrugs. So, as an overview, I'll start by talking about artificial metallonucleases before getting into some of our polynuclear compounds that we've been developing and how we can target these towards specific genomic sequences. So, I suppose the goal here, the first goal, is to design new DNA-targeted metabodrugs. And in some cases, these contain a single copper center, for example, like the TPMA. Others we've developed contain dinuclear copper centers. And in some cases, we've gone to trinuclear compounds. And we also have an interest in developing platinum drugs and some ruthenium. And so the first goal is to design new DNA-targeted metabodrugs. And the second goal then is to take these DNA-targeted metabodrugs and to hybridize or clip these to a DNA sequence. And the advantages of this approach means that we can direct DNA damage or specific interactions to a predefined sequence. And the key advantage here in terms of this technology is to use click chemistry. So some of these compounds, the copper compounds, behave as artificial metallonucleases. And we know what a nuclease is. It's an enzyme that cleaves the chains of nucleic acids into individual components. There are two main mechanisms by which nucleases operate. One is through hydrolytic cleavage. This leaves intact 3' and 5' positions, which are, I suppose, amenable to religation. Or you can develop a radical species or metal oxo species, for example, that attacks the deoxyribose or the base. And this gives a variety of breakdown products, which are much more difficult for the cell to repair. So we're interested in developing targeted oxidative DNA damaging agents. And I suppose a successful clinical example of an artificial metallonuclease is metallobleomycin. So this contains a bis-thiazole chain or bis-thiazole unit that intercalates DNA. It also has a metal binding region. This can coordinate to iron, for example, and so you now have a non-covalent interaction combined with a covalent one where you produce free radicals that can initiate DNA damage. So this is often through a cascade, so you're producing sometimes diffusible radicals, sometimes metal oxo species. But the interesting thing from an inorganic perspective is that depending on the metal complex, the shape, charge, and also the type of metal ion you use can have an influence over the position and the type of oxidative damage that you can mediate. So a few years ago, we asked a very simple question, which was the first discovered chemical nuclease by David Sigmund, shown here, copper bisphenanthroline. We wanted to know, could you introduce designer intercalators in this simple scaffold to enhance DNA cleavage activity? So we developed a DPQ, DPPZ, DPPN, and it turns out you can stepwise enhance DNA binding and cleavage activity. But one of the disadvantages here associated with these simple compounds is the dissociation of the second coordinated NN group. So for that reason, we developed these TPA, these cage systems, where in this case you've got 3 pyridyls coordinating to the copper center, but crucially there's enough room here for a designer intercalator to coordinate. It turns out these are effective topoisomerase poisons, but there's no major advantage in DNA binding affinities. In other words, the steric hindrance imposed by the TPA disadvantages the ability of the ligand to intercalate. So for that reason, we developed a simpler scaffold, DPA, which again leaves enough room for the intercalator and crystal structures. And you can see there's a stepwise enhancement in the DNA binding affinity. And in particular, this DPPZ compound has, it's a potent DNA binder and targets the minor groove, even the minor groove of GC-rich sequences. They also have significant topoisomerase poisoning activity. So moving from these mononuclear to polynuclear compounds, we work together with a number of collaborators, including Professor Tom Brown, and we can target the minor or major groove. And I want to highlight some of the work we've been doing on minor groove binders. So these systems here involve, they're relatively simple. They contain two copper bisphenolphthalein units that are, in one case, linked through a rigid terephthalate dicarboxylate linker, another one which is a flexible octane diioate. And together with the National Cancer Institute in the US, we've done screening on the NCI 60, so they're a 60 human tumor panel. They're not particularly active against leukemic cells, but in general, they have much higher activity towards solid tumors and specificity toward ovarian and some breast cancer cell lines as well. So to probe reasons for this mechanism a bit further, we took an ovarian cancer cell and incubated with antioxidants, which are specific for individual free radicals, such as L-histidine or Tyron, which scavenges superoxide, and histidine scavenges singlet oxygen. We then introduced an LD50 of the drug. And what you could find, what we're able to show is that you can significantly recover or protect the cell line from toxicity. And in particular, it seems that the mechanism is strongly linked with superoxide and singlet oxygen. And we've been able to visualize this using mitochondrial superoxide sensor, nuclear superoxide sensor, and also a singlet oxygen sensor. In terms of their DNA damaging properties, their mean tail moments, common tail moments are similar to doxorubicin. They produce a high number of double-strand breaks through the gamma H2AX assay. And in terms of their DNA binding potential, we've identified that they show intercalated profiles in AT-rich sequences. So to probe this a little bit further, we collaborated with Tom, and the interesting thing is that the binding motif or the binding interaction of this copper ODA is dependent on having a central TA-TA step. So if you have a central TA-TA step, you see this intercalated profile here. Whereas if this is reversed to an AT-AT, so identical sequence context except we're reversing the central step, we get this Z-like configuration. And so we're following this up with some additional work whereby we're able to introduce not only the AT or TA in terms of the order, but we've used diaminopurine and 1-methyladenine, and we're able to show an enhancement in the case of diaminopurine to this negative Cotton effect. So it really prefers having this diaminopurine base. Presumably there's an initial pre-associative interaction, and afterwards the base is flipped out to allow the complex to interact at the Watson and Crick phase. But with 1-methyladenine, we see no major interaction. And so the Watson and Crick interactions seem to be important along with the order of these bases. So in collaboration with Frederick Westerlund from Chalmers University of Technology, we've looked at single molecule imaging of DNA. And here we can extract primary human blood cells, introduce or expose them to our drug molecule with or without radical scavengers, extract the DNA, and then introduce base excision repair enzymes, and then fluorescently label DNTPs. And then, if these have been successfully incorporated, you end up with foci that can be imaged in single molecules of extracted genomic DNA. So it turns out that in the presence of copper ODA, we're producing a high amount of DNA damage. And when we combine this then with specific repair enzymes, you can see some have increased, we get an increase in the amount of damage detection, presumably because they're recognizing these lesions, they're cleaving them out and introducing another fluorescently modified base. And this is very different to what we find with metallobleomycin, because bleomycin is not producing very high amounts of damage in the absence of the repair enzymes. Only in the presence of the repair enzyme do we see a significant amount damage induction. And so in a subsequent experiment, we've introduced Tyron, a range of other scavengers, and you can see that we can dramatically reduce the amount of DNA damage associated. And finally, we've identified the activity in a range of triple negative human cancer cell lines, breast cancer cell lines, should I say, including this MDA-MB231, for which there are very limited treatment options. And it turns out that the copper ODA is particularly active in the low micro to nanomolar region. It's also very well tolerated by mice. You can see it's our tolerance study, and on the right we have a xenograph study showing that at a much lower concentration compared with a control carboplatin, we have a significant reduction in tumor volume associated. So this molecule has shown significant promise as a therapeutic, a new therapeutic with DNA damaging properties. So I want to move now to talk about some new scaffolds that we've recently developed, which we're calling the Tri-Clip series. So we had an idea whereby if you took a simple triazide, like this mecycline derivative, and introduced a series of amines that contain alkynes. So we've performed this reaction using a whole range of primary, secondary, and tertiary amines. Could you build scaffolds that could coordinate with copper, for example? And so using the triazole as an end donor within the scaffold, instead of click chemistry is being widely used, obviously, to join molecules together, but here it has a function. So it turns out that from the series we've developed, TC1 to TC9, Tri-Clip 1, this molecule here in the presence of copper, shows DNA damaging properties. None of the others have it. And it turns out that in the presence of copper, we get this, its DNA binding potential is significantly enhanced. So it's a potent DNA binding agent in the presence of copper. And significantly, we see a large amount of DNA damage associated with this copper TC1. We've been able to visualize the damage using atomic force microscopy. And in some cases where DNA damage occurs, we can see condensation effects from the fragments as they aggregate. We've again collaborated with Frederick Westerlund, and it turns out that DNA damage is dependent on copper. We can see scavenging activity by Tyron, and we also have specific repair enzymes that are responsible and can recognize DNA damage. How much am I doing on time?
I think you have 12 minutes.
Okay. So I think in the interest of time, I'll just go through this polynuclear platinum section quite quickly. So I think everyone should be familiar with the clinically approved platinum drug molecules, cisplatin, carboplatin, and oxaliplatin. And in recent years, to circumvent some of the resistance mechanisms, the group of Nicholas Farrell developed these polynuclear platinum compounds that can do long-range cross-linking interactions. And this is the first cisplatin, non-cisplatin chemotype to get to phase 2. And since then, there's been an interest in developing these fully transsymmetric compounds that can phosphate clamp or interact with the phosphate backbone through two limiting binding modes, GC and AT. So group backbone tracking, where the molecule interacts with a single strand of DNA, and another one across the minor groove, which is AT dependent. And one of the difficulties in tracking these molecules to give us information about how stable they are, how useful they are in terms of their clinical potential, is that if you introduce fluorophores into a polynuclear platinum compound or a platinum compound, it's going to behave much more like the fluorophore. So as a way to try and study the molecule and its biological endpoint, we thought it would be interesting to introduce a stable azide. Shouldn't interact with anything in biological media except the fluorophore, for example, on treatment afterwards. So work from Sinead O'Carroll has developed the synthetic route, which is very challenging. This is the structure of the asymmetric triplatinum C. It turns out that they behave very similarly in terms of their DNA binding activities, with one crucial difference. The interaction along a single strand, this route, this backbone tracking, is dependent on having this amine group here, which can interact with the phosphate backbone, and removing that pushes the molecule toward having a preference for AT-rich sequences. So we're modifying it so that it interacts with AT-rich regions. We've also been able to visualize some of the interactions with plasmid DNA, and it turns out that it works in terms of its intended purpose, whereby we can perform intracellular tracking. So we can identify that the molecule, once it's exposed to MDA, MD231, these are human breast cancer cells, we can then afterwards introduce fluorophores that can identify its localization, and it is in the nucleus. It's also well tolerated by mice and performs very well at a lower concentration compared with carboplatin. So the final section I want to talk about is on gene-directed metabodrugs. So we're lucky enough to coordinate two large-scale Marie Curie ITNs. The first is called ClipGene, which ran between 2014 and 2018, and the one ongoing now, Nature ETN. And so some of us have come together, remarkably, the chair, the previous speaker, the next speaker, tomorrow's speaker. And one of the goals in the network was to develop new artificial gene editing technologies. So this involves taking a therapeutic nucleic acid, performing click chemistry, and then with the idea of performing targeted DNA damage, which results in fragments that are less easily repaired. So an alternate type of gene editing technology. We have experience in developing phenanthrines, caged systems, and obviously platinum drugs. And we've managed to successfully introduce stable azides into all of these architectures and identify that they have potential as a new type of targeted technology. And I just want to highlight some of this work. I think we're running out of time quite soon. It's possible to stabilize triplexes through covalent modifications. So, for example, these intercalator intercalators introduce either the backbone or base modifications. So, with that in mind, we developed these phenanthrines in collaboration with Thomas Gurel, and crucially, these are NN groups that can coordinate to metal ions. And so, targeting the green fluorescent protein gene using a parallel TFO, which is modified either at the 5' or internal position, gives rise to, we have an ability to form stable triplexes, and in the presence of copper, we can deplete these selectively. They produce highly stabilized, they're highly stabilizing modifications, should I say, in some cases up to 22 degrees. We've also been able to make these higher order structures containing multiple metal centers that can induce oxidative damage and leave the target intact. We can also use single nucleotide incorporation primer extension technology, which was originally developed by Professor Kellett, and we've incorporated a DPA caging ligand into a DNTP and studied the activity as a targeted chemical nuclease. So this work led by Brina McGorman and Nicolau Fantoni, we could identify that the targeted DNA damage was occurring. We compared it with a synthetic and an enzymatic derivative, and they showed good selectivity. In a more complex assay using plasmids, we can see that the targeted plasmid, the green fluorescent protein sequences concentration dependent depletion, whereas the off-target plasmid, we use PUC19 in this case, remains intact. And then performing Sanger sequencing, We can identify position. So by introducing modifications on this DPA scaffold, such as DPPZ, or sorry, DPQ, DPPZ, you can enhance the position or bring the position of cleavage closer to the modification site. And finally, we've also been able to use or incorporate platinum drug molecules, and this work is in collaboration with Professor Tom Brown and his group. And for a long time, the field has been trying to introduce platinum drugs which have this cisplatin chemotype allowing a crosslink, but there's been a lot of synthetic challenges associated with doing this. Click chemistry has provided a solution whereby if you use a chelator containing an azide, you can maintain the cis chemotype. And it turns out that these you can perform targeted cross-linking with the platinum drugs. And it turns out that this interaction is on the purine-rich strand of the sequence. So hopefully I've shown you some of the work that we've done in terms of enhancing oxidative DNA damaging agents by developing new scaffolds. Polynuclear compounds have advantages, and we can also target these towards triplex forming oligonucleotides. The dinuclear copper compounds, much of this work was led by Dr. Karina Slater, Dr. Nicolau Fantoni, who's now working with Tom at ATB Bio, worked on the caged copper compounds. Rena McGorman, who's here, has worked on the enzymatic incorporation, and Joseph Hennessy on the Azido platinum analogs, and obviously to everyone who's funded this work. To you for your attention. Thank you.